Today, we are going to learn about reproduction in Cycas. In Cycas, reproduction can take place both vegetatively and sexual. Vegetative reproduction can take place by adventitious birds, bulbils, and suckers. Suckers develop from root. They grow horizontally for some time and then give rise to a new plant. Whereas adventitious bud and bulbil are found in stem. Reproduction by sucker is very common in Cycas sarcinalis. Adventitious bud and bulbil, as we discussed, they are formed in the basal part of the stem and they arise from the parenchymatous cells of the cortex in stem. The decurrent base means it is an extended leaf base which extend downward from the point of attachment along the stem and it covers the upper part of the node, which gives a wing expanded like structure. It is covered with scale leaves and from this bulbil, the foliage leaves develop which give rise to a new plant. This method is very common in Cycas revoluta. So, as we discussed about bulbil, which is found or covered with the scaly leaves, here in this diagram we can see the scaly leaves cover the bulbil and which give rise to a new foliage leaf, which finally helps in vegetative reproduction. Coming to sexual reproduction, Cycas is a heterothallic plant and is strictly dioecious, which means that a heterothallic plant where the male and female gametophytic phases, which is the reproductive phase, develop from two different kinds of spores. Therefore, Cycas produce two different types of spores, which produces two different types of gametophytic phases. This is known as heterothalism. Cycas is strictly dioecious, which means that male and female cones are present on two different plants. Either of them will be present on a single plant. In Cycas, female cones are not formed, so we may term them as megasporophylls here. So, never we can see both male cone and megasporophyll on the same plant. They will be present separately always. In India, most of the plant of Cycas sarcinalis are male and Cycas revoluta are female. Here, we can see the structure of male cone. Male cone is the reproductive structure of Cycas, which have several microsporophylls arranged along its axis. In a spiral manner. The male cone develops at the apex of the stem in between the crown of foliage leaves. It is conical, shortly stalked and compact oval structure, as we can see in the diagram. As we discussed, male cone develop at the apex of the stem in between the crown of foliage leaves. It is shortly stalked, compact, oval, conical, woody structure. One thing to be noted is, it is the largest among the plant kingdom. Cone consists of several microsporophylls arranged spirally around a central cone axis. Microsporophyll is a woody, horizontally flattened and nearly triangular structure. On the lower surface or adaxial surface of microsporophyll, there are many microsporangia present in groups. In our upcoming slide, we are going to study about microsporophyll and microsporangia. This is all about the male cone. Talking about female reproductive structure in Cycas. As we discussed earlier, female cones are not formed in Cycas. Therefore, megasporophylls are present individually. Megasporophylls are the female reproductive structure in Cycas, and these are not organized into cones. Instead, they occur in close spiral in acropetal succession around the stem of the female plant. Megasporophylls, as we discussed, that these are present in a spiral manner in acropetal succession around the stem. They have a flat and dorsiventral structure. They are divided into stalk and pinnate lamina. You can see in this diagram, there is a stalk and a pinnate flattened dorsiventral lamina. Ovules are seen on the lateral side of the stalk. We can see here this green-like structure, which are called ovules, which are attached on the lateral sides. The structure of megasporophyll and the number of ovules are different in different species and is therefore very useful for identifying the species. This was all about the male and female reproductive structure. Now, studying in deep, we've come back to the male cone. As we discussed earlier, that male cones are the male reproductive structure and they develop at the apex of the stem in between the crown of foliage leaf. They are short, compact, oval, conical, woody structure and largest among the plant kingdom. As we discussed that in male cone or strobili, there are several microsporophyll which are arranged spirally around the central cone axis. A single microsporophyll is woody, horizontally flattened, and nearly triangular structure. You can see in this diagram a triangular structure, and on the adaxial surface or lower surface, we can see in this diagram there are several microsporangia present in groups. These small groups are present and these are called as microsporangia. They are present in group of 3 to 6 and we call them as sori. In this diagram, we can see easily that the triangular structure is present where this upper sterile part is called as apophysis, whereas the lower part forms the fertile part, where on the adaxial surface there are many microsporangia present in the group and these are called as sori. There are many hairs also present in between this sori. In this diagram, if we cut the transverse section of B, we get to see this TS where this upper part is sterile part and is called as apophysis And this adaxial surface forms the fertile part and has several microsporangia arranged in group forming sori. These microsporangia have microspore mother cell which undergo meiosis and form microspores. Finally, carry out the reproduction in forward direction. Microsporangia, as we discussed, that these are arranged in groups called sori on the adaxial surface of the microsporophyll. These are sessile or have very short stalk. They are attached to the lower surface of microsporophyll and their wall is differentiated into exothecium, endothecium, and tapetum. Exothecium is thick-walled, middle endothecium is thin-walled, and inner nutritive tapetum is present, which provides nutrition to the developing pollen grains or microspores. Therefore, if we see the microsporangium, we can see the three layers: exothecium, endothecium, tapetum. There are several microspores present, which finally develops into pollen grains. Development of microsporangia. So, it is a eusporangiate type of development, which means it develops from a group of initials. Each hypodermal cell of the lower fertile surface of the sporophyll functions as a sporangial initial. As we discussed that the lower part of the microsporophyll is a fertile part. So, there each hypodermal cell of the sporophyll function as a sporangial initial. This sporangial initial divides by a periclinal division and it forms an outer primary wall cell and an inner archisporial cell. The outer primary wall cell undergoes repeated periclinal and anticlinal divisions to form 5 to 6 layered sporangial wall. The outermost layer of the sporangial wall forms the exothecium. The cell of this layer have thickenings and the archisporial cell, the inner one, undergoes several irregular divisions to form sporogenous tissue. These cells increase in size and eventually function as spore mother cell. They are the ultimate cell of the sporophytic phase, and each spore mother cell undergo meiosis by reductional division to form microspore, present in form of tetrads. They are surrounded by nutritive tapetum and which helps them to mature into pollen grains. Therefore, this is the development of microsporangia in which microspores develop. Now comes the time of dehiscence. With the maturation of microspore, axis of strobili elongates and the microsporophylls get separated. Sori get exposed and the wall of exposed sporangia dry up and break up at the line of dehiscence, releasing the microspores. The microspores are globular, uninucleate, and haploid structure, where spore wall is differentiated into outer exine and inner exine. Megasporophylls, which are the female reproductive structures. As we studied that cones are not formed in megasporophylls and they are arranged in spiral manner around the stem apex in acropital succession manner. They are flat and dorsiventral structures and are differentiated into lamina and stalk. They are considered as modified leaves and differentiated into stalk and pinnate lamina. Ovules are attached on the lateral side of the stalk and their number of ovules may vary from 2 to 10. Structure of megasporophyll and the number of ovules are different in different species and therefore it helps in identification of species of cycle. Moving forward, there are different species. Cycas revoluta have lamina very much dissected and it tapers into the point. There are 2 to 4 ovules which are arranged in 2 lateral rows. In Cycas cercinalis, lamina is ovate to lanceolate with dentate margins and acuminate apices. Their number of ovules may be up to 12. In Cycas rhomphai, there are 6 to 10 ovules. Cycas pedomae resembles very much with Cycas rhomphai, and Cycas pectinata have 4 to 6 ovules with subdolate pectinate margins. Talking about ovule. Ovule is orthotropous and unitegmic and is the largest in the plant kingdom. It consists of a large nucellus surrounded by a single integument. The integument remains fused with the body of the ovule, except at the apex of the nucellus, where it forms a nucellar beak and micropyle. Integument is very thick and it is differentiated into 3 distinct layers. Outer and inner layers are fleshy and the middle layer is hardy and stony. Mucilage canal and tenon cells are also present in the integument. The cells of the nucellar beak break to dissolve to form a pollen chamber that lies in the central region of the beak. The young ovule is green and is covered by minute cellular hairs. At maturity, it becomes red or orange color and hairs disappear. They are supplied by 3 vascular strands. They are divided into 2. One branch supplies the outer fleshy layer and the other inner fleshy layer. And this forms the complete structure of an ovule. The ovule develops from the superficial cells of the fertile part of the megasporophyll. This is the stalk of the megasporophyll where one of the superficial cells differentiate and form a small outgrowth by repeated division, forming a small protuberance. This outgrowth finally forms the nucellus by further division, and the division in the basal cell of the nucellus. These cells will give rise to an integument which surrounds the nucellus. Initially, the nucellus and integuments are free from each other, and later the integument fuses with the nucellus. The upper region of the integument forms a small aperture known as micropyle. One of the cells of the nucellus differentiates into a megaspore mother cell. This megaspore mother cell can be distinguished by larger size, dense cytoplasm, and prominent nucleus. It undergoes a reductional division and forms a tetrad of megaspores, from which 3 megaspores degenerate and only a single functional megaspore is retained, which is the lowermost megaspore, which is functional and it gives rise to a functional megaspore. This is all about reproduction in Cycas. In our next lecture, we will study about male gametophyte and female gametophyte. fertilization, and development of embryo, which completes the life cycle of Cycas. Thank you.